Good afternoon. I would like to thank Kuldev and Raj for the opportunity to speak and congratulate Gil and the organizers for another fantastic Eyecelerator meeting, including fantastic location. With that, given the focus of this meeting, or one of the foci, is the drug delivery, I would like to share with you 4 vignettes from drug delivery approaches that we utilize in development of products in the Glaukos pipeline for future therapies. And before I proceed, I would be remiss not to acknowledge terrific contributions from our clinical investigators and colleagues from US FDA, some of whom are in the room. Thank you so much. Without you, this progress would not be possible. These are our forward-looking statements and disclaimers. And so the 4 drug delivery approaches I'd like to highlight today are based on our therapeutic platforms, starting with the iDose that is capable of delivering multiple pharmacotherapies into the anterior segment for multiple years after a single dose. A second example will be based on the iLink technology, which is our technology that we utilize for collagen cross-linking for treatment of keratoconus based on bioactivated pharmaceuticals. The third example is based on our iLution platform, which is transdermal delivery via eyelid cream. And fourth example will come in from our Retina XR platform, which is our platform of biodegradable implants targeting hallmark retinal diseases, including AMD, DME, and RVO. So first example comes from iDose, and iDose is a microscopic intracameral implant that delivers multiple years of travoprost ester delivered into the aqueous humor. Here we would like to share with you 3-year data from the completed phase 3 studies, wherein we have demonstrated that iDose is capable of maintaining good IOP pressure in 70% of the patients on the same or lesser number of topical IOP-lowering medications compared to their baseline. This percentage was lesser 58% for timolol. This is underpinned by robust IOP lowering that extends into the 3rd year. And in preliminary analyses, we are showing non-inferiority to timolol at 36 months after a single dose of iDose, which is quite spectacular. This durability and robustness of the IOP lowering effect is supported further by ocular PK data. We believe this is the largest ocular PK study conducted in glaucoma patients, where we studied 210 patients. On the upper plot, you can see that the levels of travoprost ester delivered by iDose was at all times higher than the Cmax of travoprost delivered by eye drops. On the bottom plot, you can see that at 2 years after we recovered the implant, there was still 16% of the payload left, which explains the durability of the effect into the 3rd year. Now, this great safety and efficacy was greatly complemented by tolerability, wherein we had no periorbital fat atrophy adverse events, no adverse events of endothelial cell loss, very low conjunctival hyperemia, and only a single case of iris color change. So these are terrific results, and we are following this program by 2nd-generation T-Rex program that aims to deliver 6 years of IOP lowering after a single dose. So second example is for Raj, and this example has come from our treatment for keratoconus based on collagen cross-linking. In this, we had to solve a simple problem: how to deliver effective amount of riboflavin into the stroma of the cornea, how to deliver effective amount of oxygen that is consumed during the cross-linking event, and do this without disrupting the corneal epithelium. We solved this problem by generating 2 different drug product formulations that are administered consecutively and permeate the corneal epithelium to allow sufficient drug loading into the stroma. Furthermore, we have used boost goggles that deliver hyperoxic oxygen environment that replenishes the oxygen during the cross-linking. This approach was used to generate results from 2 phase 3 studies that are now submitted as a NDA review with a PDUFA date of October 20th, so stay tuned. Well, the third example will come from our iLution blepharitis treatment, blepharitis caused by infectious mites. Here the punchline is rather simple. Since the mites live in eyelid margin, the best way to target them with a transdermal delivery of therapeutics to the eyelid margin. This program will go into the clinic at the tail end of the year. And then the final example I would like to highlight is our Retina XR implant that delivers 4 years of pharmacotherapy in the preclinical models and is currently studied in an ex-US phase 1, phase 2b study that will be moving into the United States, given by the IND being opened 3 days ago. So given this great progress in drug delivery approaches in our pipeline, we are truly excited and executing on our strategy to fully leverage the value of drug delivery and making a difference in glaucoma and ophthalmic patients worldwide. Thank you so much for your attention.